Now primarily we will be focusing on restriction enzymes and how they get cut. Whenever we take a restriction enzyme like ECOR1 or HIND2, they can recognize a specific sequence of the DNA within the bacterial cell, and they can recognize a sequence which is termed as a palindromic sequence. What could be a possible palindromic sequence? If you take the DNA, on one strand it has 5', it has got GAATTC 3'. On the opposite strand of the DNA, because DNA shows the opposite polarity, we have another strand which is 5', and the same sequence occurs in the form of a mirror image. It occurs in the form of GAATTC 3'. If you look at GAATTC on one strand and GAATTC on the other strand, both of these sequences are just the same but oppositely oriented on both the strands, and such sequences are termed as palindromic sequences. Like if you recollect the word of little, if you can read the same on both the ways, or if you recollect a sort of a phrase saying "Madam, I am Adam," it reads the same. So palindromic sequences are specific sequences. They are specific sequences which read the same on both the strands. Their location of the palindromic sequence, the frequency of occurrence, the number of times it occurs, the palindromic sequence is species specific. Is specific for the sort of a species of a bacterium or a microorganisms. So if you take Escherichia coli, or else you are taking Erwinia carotovara, or else you are taking Streptomyces, in each of these particular strains, the palindromic sequence which are present on the DNA are specific sequences which are in turn recognized by restriction enzymes. Now what do palindromic sequences convey? The palindromic sequences conveys that it is a recognition sequence and it is recognized by the specific battery of restriction enzymes. Now, whenever we have a restriction enzyme, the restriction enzyme has a specific pattern of cutting first recognizing the palindromic sequences, and next it cuts or cleaves or knives or scissors, or it is used as a sort of a molecular, the restriction enzyme is also termed as molecular scalpels. The reason why they are called as molecular knives or molecular scalpels is they recognize a specific sequence. Like if you take the sequence of GAATTC, and if we have a restriction enzyme called ECOR1. The ECOR1 recognizes a sequence and it recognizes the palindromic sequence of GAATTC and CTTAG. Number one, it recognizes the palindromic sequence. Next, it recognizes what sort of a sequence? It recognizes a 6-cutter sequence. Remember, we got GAATTC is termed as a sort of a 6-cutter. And always ECOR1 cleaves or cuts the 6-letter sequence between G and E. Between G and E. If you look at the sort of an arrows which are put in the opposite direction, these indicate the places where the sort of a restriction enzyme cuts the sequence. So it is a 6-cutter based sequence. It cleaves between G and E. And whenever it cleaves, the restriction enzyme cuts the sequence along the dotted lines. If you just look this way, 1, 2, 3, 4, and it cuts along this set and along again on the set. So we understand that restriction enzymes cut both the strands of the DNA. They cut both the strands of the DNA. So when it cuts both the strands of the DNA, you understand there is a sort of an L-shaped structure which comes in. And if we are looking in terms of restriction enzymes and the pattern of their cutting, always when it cuts, normally it cuts by two important methods. One is termed as a sort of a blunt cut. A blunt cut is one wherein, just like how you shoot an arrow through a nucleotide sequence, it recognizes polarity and correctly cuts the sort of a DNA sequence into equal halves. So this is termed as a sort of a blunt cut. A blunt cut is a sharp cut which exactly goes straight through the sequence and it recognizes the sort of a symmetry of the sequence and cuts it into equal halves. But most of the restriction enzymes, which are very important for molecular biology, which are very important tools in molecular biology, they follow another pattern of cutting which is termed as a staggered cut. So among the cutting sequences, we have a blunt cut and we also have a staggered cut. So in a staggered cut, it will look like as if you have taken a sort of an instrument and sawed a piece of wood. When you saw a piece of wood, the wood comes off in the form of a sort of a chip. So in a staggered cut, we always have a sort of a sticky or a sort of a cut end. It is just as if you have the sequence and the sequence also has got one sort of an edge on the outside. So when you have a restriction enzyme which is cut like this, because of this particular cut, we have AATT and also C. And similarly, we have AATT and C. So this one comes, this AATT and TTAA is termed as a sort of an overhang. That is, it comes on the upper surface, which is called a sort of an overhang. So whenever we have a staggered cut, a cut which is abnormal, a cut which does not cut the sequence correctly at the center, but it shears through the particular sort of a cut, the enzyme, it produces a sort of an overhang. And normally the overhang which is developed, which is formed due to ECORI, ECOR1 is TTAA. And this sort of an overhang, because it has got an overhang, these ends, that is AATT, what they could be is, they just act just like adhesive tapes. They act like gum tapes and they are sticky. First thing is, it is sticky, it is called a staggered cut and they can anneal. So when you have your sort of a DNA piece, the DNA piece has got an overhang and these overhangs are just like tapes, like adhesive tapes, and they can anneal, they can rejoin. So because they are made up of gums, they are just like gum-like or they are adhesive, they can anneal and go to form rounded molecules, and when they form rounded molecules, it is because of the presence of cohesive ends. The meaning of the term cohesive ends is joining ends. Thereby we understand when we take restriction enzymes, when we cut restriction enzymes, the restriction enzymes can again come together and it can go to form a sort of a circular molecule. It can go to form a circular molecule because they can join, they can ligate, they can anneal, and thereby you can take a linear stretch of restriction enzyme, you can provide staggered cuts or sticky cuts, they have got a sort of an overhang, the overhang can anneal, and in the presence of another series of enzyme which is also called a sort of a tool which is termed as a DNA ligase. DNA ligases are enzymes which act as gums. DNA restriction nucleases act like scissors. DNA ligases act like gums. So they can anneal, they can adhere, they can join, they are coherent, they are sticky, and they can enable a linear molecule of DNA to become a circular molecule of DNA. Thereby, we understand that whenever we are studying on restriction enzymes, the basic features of restriction enzymes are they are sequence specific. It has got a palindromic sequence. Restriction enzymes can recognize a palindromic sequence. It can lead to a sort of a 4-base or a 5-base or a 6-base cutter. It can provide a sort of a blunt cut or it can provide a staggered cut. When it forms a staggered cut, it can go to form cohesive ends. And when there are cohesive ends, the cohesive ends can anneal. When the cohesive ends anneal, a linear molecule is converted into a sort of a circular molecule, and the circular molecule by itself can load in, or within the circular molecule you can have the input of a sort of a foreign gene, and the circular molecule can be sent from one bacteria to another bacteria. So this is primarily about the nomenclature and the cutting sequence of restriction enzymes.